Hello, everyone. Today we're going to be talking about contingency analyses. A contingency analysis is just a way to estimate and test for an association between two or more categorical variables. And what I mean by an association is that the two categorical variables are not independent of one another. They're somehow associated. So for example, we might test for an association between whether or not an individual died in the sinking of the Titanic and whether they were a man or a woman. Here we've plotted the data using what's called a mosaic plot, where the size of the box represents the number of individuals in that category. So we can see, first of all, that there were many more men on the Titanic than women. That's why this bar is thicker than this bar here. And you can see that also by the numbers in these cells. But in addition to seeing that there were more men than women on the Titanic, we can also see that men were far more likely to die when the Titanic sunk than women were. This big red bar is much bigger than the small yellow bar corresponding to the men that survived, whereas the proportion of females or women that survived was much higher. So in this case, an association between whether or not an individual died and whether they were a man or a woman is indicated by different heights or different proportions of these red and yellow bars. If these two categorical variables were independent of one another, we would expect to see similar sized red boxes and similar sized yellow boxes. As another example, as a master's student, I tested whether dietary constraints could limit reproduction in female deer mice. And so I did a comparison of the number of females that bred as young of the year, meaning in the summer of their birth, in control plots, plots that were supplemented with high energy but low protein food, shown by E here, and plots that were supplemented with high protein food. And you can see from these bar plots that the proportion of females that bred was much higher in the protein supplemented populations than in the controls or the energy supplemented populations. So in this case, then, I could potentially have used counts of the number of females that bred or didn't breed in each of these three scenarios to test whether there's an association between diet and whether or not females bred during the summer of their birth. So here's what the data looked like. There were 3 females in the control population that bred as young of the year and 4 that did not. 2 in the energy supplemented population that bred and 4 that did not. But in the protein supplemented population, all 5 young of the year females bred. And this is then depicted in this mosaic plot again. In this case, the totals for controls, energy, and protein were relatively similar. So the widths of these columns is relatively similar. But you can see that the relative height of the yellow and red bars is really different across these 3 treatments. The proportion of females that bred on control and energy supplemented study areas was relatively similar, but very different from the protein supplemented population where all of the females bred. Now, for most of the remaining part of this video, we're going to focus on an example of a really interesting experiment trying to test whether infection with a trematode parasite in the brain of killifish influences their susceptibility to predation by birds. Now, parasites do all sorts of amazing things, in part because they spend part of their life cycle in one host, but then in order to reproduce, need to get to a second or definitive host. And so parasites somehow have evolved remarkable abilities to control the behavior or influence the behavior of their intermediate hosts as a way to make those intermediate hosts more susceptible to predation by their definitive hosts. And one example of this is this trematode that was studied in this particular study. So the researchers wanted to then test whether infection by this trematode in the killifish made them more susceptible to predation by birds. So what this study by Lafferty and Morris tested then was the hypothesis that infection by this parasite influences the risk of predation by birds. So they used an outdoor tank that was stocked with 3 different kinds of killifish: those that were unparasitized, those that had relatively low levels of parasitic infection, and those that were heavily infected. The tank was then left open to predation by birds, and the researchers counted how many of each of the fish in these 3 different categories were eaten by birds. And this is what the data look like. You can see that there were a total of 50 uninfected fish, of which only 1 was eaten. There were 45 lightly infected fish, and 10 of the 45 lightly infected fish were eaten. And in total, there were 46 highly infected fish, and 37 of those 46 highly infected fish were eaten by birds in this experiment. Now, here's what the data look like shown as a mosaic plot. And we can see that very few of the fish in the uninfected category were eaten, whereas a really high proportion of fish in the highly infected category were in fact eaten by birds. The fish in the lightly infected category fell somewhere in between, but it's clear that the heights of these yellow and red bars in each of the 3 categories are not the same. So this mosaic plot then suggests that there is in fact an association between infection with this trematode and whether or not the fish will be eaten by a bird predator. So in this particular example then, we might want to test a null hypothesis that parasite infection and being eaten are independent of one another. In contrast, we might use the alternate hypothesis that parasite infection and being eaten are not independent of one another. And this style of null and alternate hypothesis is really characteristic of contingency analyses. The null hypothesis typically indicates that the categories are independent of one another, whereas the alternate hypothesizes that they are in fact not independent of one another. These are not really very sophisticated alternate hypotheses. We're not really making any predictions about which levels are higher or lower than others, just that they are not independent of the other category that we're looking at. Okay, so the mosaic plot suggests that perhaps there is an association between trematode infection and whether or not the fish are eaten, but we would naturally expect some differences to occur just by random sampling error. So we need to perform some sort of statistical test to determine whether the differences that we're observing or the associations that we're observing between infection and whether or not the fish are eaten are really unusual compared to what we would expect just by sampling error if the null hypothesis were true. So are these differences greater than what we would expect just from sampling error? So to test this, we need a test statistic. And the test statistic that we use for a contingency analysis is one that we're already familiar with from a goodness of fit test. And in fact, a contingency analysis is just a special case of a goodness of fit test. So in this contingency analysis, then, we're going to use a chi-squared test statistic just like we did before, where we calculate the chi-squared test statistic as the sum of all squared differences between observed and expected values divided by the expected values. But one important difference then between a goodness of fit analysis and a contingency analysis is in how we calculate these expected values. If you recall in our goodness of fit test, we calculated our expected values by using some null probabilities or proportions associated with each of our categories. Well, to do this for a contingency analysis, we need to consider both of the categories at once. So for example, the 3 different categories of infection and the 2 different categories of whether or not the fish were eaten. So the first step we want to take then in calculating our expected values is to sum up the number of observations in each row as well as in each column. So in the first row, we have 1 plus 10 plus 37 equals 48, whereas the number of fish that were not eaten across the 3 different infection types is 49 plus 35 plus 9, totaling 93. We can see that there were a total of 50 uninfected fish, 45 lightly infected fish, and 46 highly infected fish, for a grand total of 141 fish in this experiment. So the next step in calculating our observed values is to try and determine how many observations we would expect to see in each of these cells if the null hypothesis were true. Now, if you recall from our video on probabilities, you'll know that when the probability of infection is independent of the probability of being eaten, then we can calculate the probability of a fish being uninfected and being eaten as the probability of it being uninfected multiplied by the probability of it being eaten. And this is exactly what we want to calculate. We want to calculate this probability assuming that these 2 processes are independent of one another. All right, so how are we going to calculate this? Well, remember from our probabilities, the probability of being uninfected is simply the number of observations of fish that are uninfected divided by the total number of instances that occurred. So the probability that any fish in the experiment was uninfected was 0.3546, or 50 divided by 141. Now, we can also calculate the probability of being eaten then as the number of fish that were eaten out of the total number of fish in the experiment, which calculates out to be 0.3404. And I think you can tell that this is exactly why we calculated the totals across the rows and columns so that we could calculate these probabilities. So we can calculate then the probability that a fish was uninfected and eaten as the product of these 2 probabilities and calculate that that overall probability is 0.1207. So if the probability of a fish being uninfected is independent of the probability of it being eaten, then the probability of it being uninfected and eaten is the product of these 2 separate probabilities. Okay, now these are the probabilities, but how do we get the expected number of fish in this cell? Well, to get that, we simply take the probability of fish occurring in this cell and multiply that probability by the total number of fish in the experiment to get the expected number of fish in this category. So in this experiment then, if being eaten is independent of infection status, we would expect to see 17 fish that were uninfected and in fact eaten by the birds. So we can follow a similar calculation for all of the other cells in this table to come up with expected values for all 6 of the combinations of infection status and whether or not the fish were eaten, which would end up looking something like this. Now, before moving further with our calculations, it's worth pausing for a minute to check some of our assumptions. Now, because the chi-squared contingency analysis is just a special case of a chi-squared goodness of fit test, then the same assumptions apply. We assume that our sample is a random representation of the overall population. Also, that none of our expected values are less than 1, and no more than 20% of our expected values are less than 5. So if we revisit this table of expected values, we can see that none of the expected values are less than 5. And so if the sample was collected randomly, the assumptions of our chi-squared contingency analysis test have been met. So since the assumptions of our analysis are appropriate, we can move ahead and calculate our chi-squared test statistic for our contingency analysis. Now here I've simply expanded out the summation across both the rows and the columns. But basically what we're doing is adding up all of these values across all of the cells in the table. So for each cell, we take the observed value, subtract the expected value, square that difference, and divide that squared difference by the expected value. Okay? And so we can do that for each of the 6 cells in our analysis, as I've shown here. So in our table, we had 1 observation in the first cell, but the expected value, if these processes are independent of one another, is that we would have had 17 observations in that cell. So observed minus expected squared divided by expected. And we do that for each of the 6 row by column combinations in our table. And if we plow through this and do all of the math, we would calculate a chi-squared test statistic of 69.5. So if you recall from the goodness of fit video, the chi-squared distribution depends on its degrees of freedom. So the probability of getting a chi-squared value of 20 really depends on whether we're talking about a distribution with 5 degrees of freedom, 11 degrees of freedom, or 20 degrees of freedom. So it's really important then that we calculate and remember and report our degrees of freedom, because the probability associated with a test statistic is really going to depend on those degrees of freedom. If you remember in our chi-squared goodness of fit test, we calculated the degrees of freedom as the number of categories minus 1. In our chi-squared contingency analysis, the calculation of the degrees of freedom is a little bit different. In this case, the degrees of freedom are equal to the number of rows minus 1 multiplied by the number of columns minus 1. So in our example, we had 2 rows and 3 columns. So 2 minus 1 multiplied by 3 minus 1 is equal to 2. So in our example of the fish being eaten by birds and being infected by trematodes, the degrees of freedom in our chi-squared analysis is 2. So we can then assess whether or not our calculated chi-squared value is unusual, either by considering a critical value or by calculating the exact probability from the chi-squared distribution. So if you recall, a critical value then is the value of the test statistic for which the p-value is equal to our alpha. So for a chi-squared distribution with 2 degrees of freedom, a chi-squared test statistic of 5.99 will have a p-value equal to 0.05. And so the notation here is our chi-squared test statistic with an alpha of 0.05 and 2 degrees of freedom has a value of 5.99. So if our calculated chi-squared test statistic is greater than this critical value, then we would reject the null hypothesis. And if our calculated chi-squared value is less than this critical value, then we would not reject the null hypothesis. Now, these are the critical values, but we can just as easily calculate the p-values directly from this chi-squared distribution. So here I've drawn a chi-squared distribution with 2 degrees of freedom, where I've indicated the critical value here, and this shaded red area then represents 5% of the total area under this curve, or the value of 5.99 has 5% of the distribution that falls to the right of it. So what was our calculated value again? Well, if you remember, we calculated our chi-squared test statistic with 2 degrees of freedom from our observed sample to be 69.5, which is pretty much off the charts in terms of this chi-squared distribution. There's only a very tiny chance that we would get a chi-squared value like this if the null hypothesis were in fact true. And we can use some code in R using this p chi-squared function, specifying our test statistic, our degrees of freedom, and that we want the upper tail of the distribution. And we see that that value is very small, 8 times 10 to the minus 16. So what can we conclude then? Well, our test statistic was very large relative to the critical value for a chi-squared test statistic with 2 degrees of freedom. And so the chances of getting a test statistic as extreme or more extreme than what we observed in this experiment is really, really, really low. So the more parsimonious conclusion then is that the null hypothesis is in fact false. So in this case then, we reject the null hypothesis that trematode infection is independent of predation by birds, and instead suggest that there is some association then between trematode infection and whether or not the fish are eaten by birds. Now we need to be a little bit careful because an association between these two variables does not necessarily mean that infection causes the fish to be eaten. In this case, it's a little bit hard to imagine that causation was the other way around, where being eaten by a bird or not caused the fish to be more or less infected. That seems a little bit odd in this particular experiment. But it is always possible that this association could be caused by some other underlying relationship or variable, sometimes called a lurking variable, that is in fact responsible for this relationship. But it's important to remember in any contingency analysis that we're always simply looking for associations. There's never any implied direction or cause of the associations. It's also important to remember that in a contingency analysis, we don't differentiate which categories are different from which other categories. Now, in a 2x2 contingency analysis, this would be pretty straightforward. But in this case, we had a 2x3 contingency analysis. And we don't really know whether this overall lack of independence between infection and whether or not the fish was eaten is the result of differences between the uninfected category and the low category and the high category, or the high category and the uninfected category. So in this example, then, we know that these two variables are not independent of one another, but we don't really know where those dependencies or associations are. It's also important to consider our assumptions. If we go back to my example of my study of deer mice, where I was looking at whether or not the female deer mice bred during the summer of their birth in relationship to these two treatments and one control area, you can see that the observed numbers or counts in each of these categories is really pretty low. And if we calculate the expected values for each of these 6 cells, you can see that in all 6 cases, the expected values are less than 5. So in this case, it would be not wise to proceed with a chi-squared contingency analysis. So when we have expected values that are too small, what can we do? Well, one option is to perhaps combine categories, but it's important to note that in some circumstances, this won't make any sense. In other circumstances, it might be reasonable, and this might be a reasonable way to proceed. When this doesn't make sense or is not possible, or perhaps even in combining categories, the expected frequencies are still too small, then we can use other approaches such as a Fisher exact test or a permutation test. Now, we're not going to go into either of these tests today, but I did want to point out what these options are to you in case you come across a situation where your expected values are too small. So in summary then, in this video, we've talked about contingency analyses and talked about how a contingency analysis is a test for an association between 2 categorical variables. The test statistic that we use in this case is a chi-squared test statistic, just like we used for a goodness of fit test. But in this case, we calculate the expected values for each of our cells by looking at how many observations we had in each row and each column, and we calculate what the probability and the expected frequencies are in each cells on the assumption that these 2 processes are independent of one another. As with any test statistic, we then want to compare our calculated test statistic with a distribution of test statistics that we would expect to get if the null hypothesis were in fact true. So in this case, we compare our calculated chi-squared value with a chi-squared distribution that has a degrees of freedom equal to the number of rows minus 1 multiplied by the number of columns minus 1.